Good morning, students. Today we will be starting with a new topic, excretory products and their elimination. Now, to begin with, what is excretion? We define excretion as the removal of metabolic waste from the body. When we talk of metabolic waste, we have to use the term nitrogenous waste. So, the right definition of excretion will be the removal of metabolic waste, mainly nitrogenous in nature, from the body. Now, the food that we eat is digested, it is absorbed by the blood, and then it is distributed throughout the body. When this food is broken down in the tissues, many toxic and poisonous substances are produced. These have to be removed because they may be fatal if present beyond a certain amount, beyond a certain concentration. And most of the metabolic wastes are nitrogenous substances which are produced by the metabolism of amino acids. That is why we say excretion is the removal of metabolic wastes, particularly nitrogenous in nature, from the body. Then there is another important term about which we should know, that is osmoregulation. Osmoregulation is the process by which the water content and iron concentration is regulated in the cells. By this, the osmotic pressure in the blood and the tissue fluids is kept constant. And this process of osmoregulation and excretion together bring about homeostasis in the body, and so they are interconnected. And in higher vertebrates, like we are talking about mammals, about humans, these two processes are carried out by the kidneys, because kidney is helping in removal of nitrogenous waste and is also helping in maintaining water balance in the body. And this is what we are going to see, how the kidney functions and how excretory products are eliminated, and the homeostasis can be maintained. Steady state of all the substances in the body are maintained. The constancy is maintained by the kidneys in the body. So, basically, the major excretory wastes that are produced in the body are carbon dioxide, which is released during aerobic respiration, then bile salts and bile pigments, which are produced due to the breakdown of the heme part of old worn out red blood cells in the liver. The bile pigments that are produced are biliverdin and bilirubin, and the bile salts are produced during lipid metabolism in the liver. Mainly the nitrogenous substances like ammonia, urea, and uric acid, they are formed during the metabolism of proteins and nucleic acids. And then water is also an excretory product. Extra water has to be removed out from the body, and water is produced as a byproduct in number of reactions, and even during aerobic respiration. So this is in general about excretion, osmoregulation, the different kinds of excretory products, but the main excretory product is urea in humans. Besides this, other nitrogenous wastes produced are uric acid and even ammonia. And then this was about humans, but nitrogen excretion varies in different animals, and basically nitrogen excretion depends upon the availability of water to the organism and its habitat, then the kind of enzymes that are present in the body, then it is also dependent on the mechanism of water loss control that is present in the body. So animals mostly excrete excess of nitrogenous waste as ammonia, urea or uric acid, and according to the principal nitrogenous waste products that are excreted, animals can be of the following three kinds: either ammonotelic, those that excrete ammonia, and the mechanism involved is referred as ammonotelism. Then animals can be ureotelic, mechanism involved is ureotelism, that is urea is the main excretory product in such animals, or they can be uricotelic and the mechanism involved is uricotelism and the main excretory product is uric acid. So we will be talking about each of these now. To begin with, ammonotelism. Ammonotelism is elimination of nitrogenous waste in the form of ammonia, and during the breakdown of protein, during the catabolism of proteins, deamination takes place and it results in the formation of ammonia, which is the basic nitrogenous waste product. So ammonia is removed from amino acid and ammonia is highly soluble in water. So as it diffuses rapidly in water, it is highly toxic and so it cannot be stored in the body for long. It has to be eliminated as soon as it is formed, since the concentration of ammonia is maintained at just 0.2 mg per 100 centimeter cube of blood, which becomes toxic. And such animals are generally aquatic in nature that excrete ammonia, because large amount of water is required to eliminate ammonia. So ammonotelism is suitable for aquatic animals like the animals of kingdom Protista, all aquatic invertebrates, then fishes, freshwater fishes, and the amphibians in their larval stages, because they are found in water. Some animals, this is a point to be noted, some animals like earthworms, they excrete ammonia only when there is plenty of water, and they are at that time termed ammonotelic, but they turn ureotelic, that is excrete uric acid when the environment is dry. So in drier animals, in drier environment, earthworm becomes ureotelic, excretes urea, and otherwise when there is plenty of water, as in the monsoons, the earthworms are ammonotelic in nature. They excrete ammonia. Similarly, the toads and lungfish are ammonotelic in water, and they turn ureotelic when they lie dormant in moist mud during summer months. So this was about ammonotelism, where the nitrogenous waste excreted is in the form of ammonia. Then we come to ureotelism. Ureotelism occurs in animals which cannot eliminate ammonia rapidly. As I told you, ammonia needs a lot of water to be excreted out. So if water is not available, ammonia is readily converted into urea by combining with carbon dioxide in the liver. And in this reaction, in this process, a large amount of energy is required so that ammonia can convert to urea. And urea is less toxic than ammonia, and it can be stored in the body for a longer time. Urea is also soluble in water, but less than ammonia. So it can be eliminated when sufficient water is available, as in aquatic animals, or it needs to be concentrated in the urine, as in most of the terrestrial animals. And examples of these ureotelic animals are all mammals, whether they are terrestrial or aquatic. So aquatic mammals like whales and seals also eliminate urea as their excretory nitrogenous waste. So all mammals are ureotelic animals. Even the desert animals like the camels, rats, kangaroo rats, these animals that are found in desert also excrete urea as their nitrogenous waste. Then certain reptiles like alligators and turtles, certain aquatic amphibians like toads and frogs and cartilaginous fishes, they also excrete urea as their nitrogenous waste. Now another thing, man concentrates urea, humans concentrate urea in the urine to more than 70 times its concentration in the blood. And so we have talked about ureotelism and the animals that excrete urea as their nitrogenous waste. Then the third category is uricotelism. As I have already told you earlier, uricotelism means animals that excrete uric acid as their nitrogenous waste. Now the ammonia that is produced, it is converted into compounds like potassium urate, and then they are converted to uric acid in the liver of higher animals. And again, here in this process, a large amount of energy is required, even more than that is required for conversion of ammonia to urea. So conversion of ammonia to uric acid needs a lot of energy. Uric acid is least toxic and almost insoluble in water. It can be stored in the cells or even organs for a long time without producing any harmful effects. Uric acid can be stored for a very long time, and for the excretion of uric acid, very little amount of water is required. So, uric acid becomes the most suitable excretory product for animals that are adapted for terrestrial life, and basically animals that have shelled eggs and the birds, they are well adapted for excretion of uric acid as their chief excretory product. Now, in mammals, the soluble wastes like ammonia and urea pass out into the mother's blood during the developmental stages in the embryo. However, in case of shelled animals and shelled eggs of birds and other reptiles, there are fine pores present which are permeable to gases only. So, if a shelled egg produced ammonia or urea, these wastes would require a lot of water, and these soluble wastes then would have accumulated in the egg and become fatal, become toxic. So, they are for excretion of nitrogenous waste, the ammonia is converted to uric acid, and then uric acid gets precipitated out of solution and is not toxic. It is stored in the shell as a solid waste which is left behind when the egg hatches, animal comes out of the egg, and this waste, uric acid, because it is not toxic, does not harm the animal. And in case of birds, birds do not have a urinary bladder, and the urine passes out by way of ureters. Uric acid being insoluble, the water is almost completely reabsorbed from it, and uric acid and the solid excretory waste, that is the fecal matter, they are discharged together from the cloaca as a semi-solid or in the form of a paste. And we have often noticed that in the bird droppings, the white matter is the uric acid, whereas the other grayish blackish matter, solid matter, is the fecal matter. And uric acid concentration is so high in the bird droppings that uric acid is also removed from the bird droppings for commercial use. So this was about the 3 excretory mechanisms that are found in animals. So basically this was an introductory class today about the different kinds of excretory products that are produced in animals, and on the next turn we will be discussing the human excretory system in detail. So take care. Good luck to all of you.